Classic Punnett squares. Gregor Mendel, the Austrian monk who grew peas and gave us our modern understanding of the gene and genetics. So, if you would take, like Mendel did, a yellow round pea plant and cross it with a green wrinkle plant, what would their offspring look like, Lucas? Round and yellow, and that's what we see right here. Now, why is that? Why would they all be round and yellow? It's dominant. Yeah, will they always get a dominant allele? So, this is one parent, big R, big R, big Y, big Y. All their kids, all those baby pea plants are going to get a big R and a big Y, all of them. So they're all going to show that dominant trait. This parent's going to just contribute the recessive version, right? So all the kids are going to be have one of each and be heterozygous for both traits. Okay, it will be round and yellow. Each plant will get a dominant allele. All right, Nico, what if you take those kids, those baby plants, and they grow and they get mature and then they have baby pea plants on their own? What's going to happen then? What will those offspring look like? Anything you can see in the graphic? Will you get round and yellow ones? Yeah, you'll get round yellow. Will you get some other ones that look different? Like Nico, what do these ones look like? How would you describe those? Well, are they round? No, they are wrinkled. But what color are they? Yellow. And how would you describe, these ones? Round and green. So, round and green. And is that all we got? Do we have any others? Do we have just those three phenotypes? Let me use that word. You guys know what the word phenotype means? Phenotype versus genotype. That'd be a good word to. Any guesses on that? Annabelle, any guesses on that? It relates to what we're talking about with, we're looking at the alleles, the genes something has, and what their appearance is. So what could phenotype or genotype mean? Any guess? What does genotype sound like? Well, genetic, yeah. And type is like that kind of categorize something, right? So genotype that means the genes, or, alleles, because we use that interchangeably. The genes or alleles an organism has. So whatever their genes are. Can you tell the genes that a living thing has just by looking at it? Is that possible? Oh, thank you. Like if I look at you, can I see all of your genes? Can I tell what your genes are? No, you can't. Can you sometimes, though? Can you sometimes figure out what genes somebody has? Oh, Emily, you put that gum in the garbage, please. It depends, yeah. So the answer, it depends almost always. If somebody remember, we don't all get a dominant allele for something. If you get 2 recessive alleles, you'll show a trait that's different, right? Like this wrinkled trait. If you're wrinkled, are you going to have any big R's? No, you're going to have little r's. So I would know what genes you have if I see that you're wrinkled. But if you're round, do I know, what could you be if you're round? Will you have to have at least one big R? Yeah. But the other R could be big R or little r, right? So I couldn't tell in that case. So genotype, usually you have to do some test crossing or, look molecularly. Now the word phenotype, that has to do with not the genes you have, but your appearance or the appearance of the living thing. I'm just going to put the appearance. So when we've been talking up here, we've been talking about phenotypes, right? Round, wrinkled. And we've been talking about genotypes, big R, big Y, little y, and so forth. All right. So we have 3 phenotypes. Are there any other phenotypes there? Are those the only ones? How about Jacqueline? Do you see another phenotype? Let's look at a bigger one. Can you see? Let me write that again. Let me write that down. We got round, phenotypes. Round, yellow, wrinkled, yellow. We got round, green. Any other appearances? Wrinkled, green. Yeah, we got wrinkled. Are there any other phenotypes? That's it. Okay. Now, let's look at let's write down the genotypes. That wasn't on the question, but let's do that. So what's one genotype we have? Big R, big Y, big Y, big R, little r, big Y, big Y. Is that a new genotype there? I think it is. Big R, big Y, little y. And here's my heterozygous. You'll see, yeah. Okay, is this a new one here? We have that already. We have that already, right? What about this one? Is this new? I think it is, right? A little r, big Y, big Y. Is that a new one? No. Is that a new one? Yes. Little r, big Y, little y. Okay. Is that a new one? I think we have that already, right? Got right there. That one we have. Ooh, is this a new one? Yes. Big R, little y. And that's the same, isn't it? That's the same as this one. That one we had before. Ooh, is that new? It is not. We had it before. What about this one? Two little y's. That's a new one, isn't it? Big R, little y, little y. And is this new? Yeah, we don't have that. So little r, little. So how many genotypes do we have? We have 4. 1, 2, 3, 4, 5, 6, 7, 8, 9. So out of 9 combinations of alleles, we can get in this case, 4 different genotypes. Okay? So just make sure, you practice, understand those terms, genotypes and phenotypes. Now, let's look at how many round yellows are there. Count them all up in there. How many round yellow? Ooh, uh-oh. Oh, there we go. Oops, that's not what I want. There we go. How many round yellows are there? 9. So, how many wrinkled yellows? 3. How many round green? 3. How many wrinkled green? 1. So this is a classic ratio. You guys that did your biozone work, if you cross 2 homozygous, if you cross 2 heterozygous individuals, you'll always get these 9:3:3:1 ratios. Okay, that's kind of the magic ratio. If you do not get that ratio, or if you get ratios that are not what you expect, that means something's going on. It means genes are linked or there's evolution happening. But remember this 9:3:3:1 ratio when you cross 2 heterozygous individuals. Okay, I'm pretty sure that's going to come up somewhere on the exam. That's kind of a real classic. This is the classic Mendelian, dihybrid cross ratio. So write that down in your paper. Dihybrid cross expected ratio. Okay? All right, hopefully you can fit most of that on your paper. All right. Today I want to finish up your our chromatography experiment. So get out all your paperwork, all your stuff on that. I'll leave this up here for another minute or two. Including, that secondary data table you wrote down those values on. Get that out as well.